Hello friends, welcome back to my YouTube channel Nursing Padasala. This is a part 3 video. Currently we are revising for a BARC OCS examination. First question is the molecular weight of protein is 30 kilodalton. The minimum length of polypeptide encoding this polypeptide will be what? That is, they are asking what is the minimum length of messenger RNA. So first they had given molecular weight of protein which is 30 kilodalton. First we need to convert the kilodalton into dalton, which means you need to multiply this number with 1000. Okay, so the molecular weight of protein will be 30,000 dalton and the molecular weight of one single amino acid is 110 dalton. So now you need to find what is the total number of amino acid present in the polypeptide. Okay, so what is the total? How you need to now find the number of amino acid, which is weight of protein divided by weight of one amino acid, which simply gives the total number of amino acid. So 30,000 divided by 110, which means totally 273 amino acids are present in the polypeptide. So from this number, we can able to easily calculate the length of messenger RNA. So length of messenger RNA is equal to number of amino acid multiplied by 3. So how you can able to get this number 3? So if you take about the messenger RNA, it is made up of nucleotide bases and 3 nucleotide will be coding for. So here you can able to find 3 nucleotide bases in mRNA will be forming one codon, and one codon is equal to one amino acid. That's why you need to multiply 3 along with the number of amino acid, which is 819 nucleotides are present. That is the minimum length of polypeptide, which is equivalent to 800. Okay, so if you want to find the number of amino acid means, then this is a formula. If they are asking, if they had given a length of mRNA and if they are asking to find the number of amino acid means, it is number of amino acid is equal to length of messenger RNA divided by 3. For example, in this example, a prokaryotic gene has 600 nucleotide long. How many amino acid will this particular polypeptide gene will be encoding? So here they are asking to us to find the number of amino acid. So number of amino acid is equal to length of mRNA, which is 600 divided by 3, which is 200. Okay, so how many amino acid will be there? It is 200. Next question, a messenger RNA is 336 nucleotide long, including initiator codon and terminator codon. So they had included the initiator codon, that is start codon, and the termination codon, that is stop codon. Okay, so initiator codon codes for the amino acid called methionine, but this termination codon doesn't codes for any amino acid. So you need to note on that particular point. Find the number of amino acid in a polypeptide chain that can be translated from the messenger RNA. So first we need to find the number of amino acid. So number of amino acid is equal to length of messenger RNA divided by 3, that is 336. That is the number of nucleotide divided by 3, which is 112. So here they are asking how many number of amino acid in polypeptide chain can be translated. So now you need to minus 1 from this 112. Okay, so the correct answer is 101. Why we need to minus this 1? Okay, so here as I mentioned earlier, the start codon will be coding for the methionine, but the stop codon doesn't code for any amino acid. It's just a termination codon. Here they are asking how many amino acid can be translated to a protein, like that they are asking. That's why you need to minus this one. This one is a stop codon. Okay, if they are asking us to find the number of amino acid alone means no need to minus this 1. But if they are specifically asking you to find the number of amino acids in a polypeptide chain that can be translated from the messenger RNA means, definitely you need to minus 1. That is a stop codon. Stop codon doesn't code for any amino acid. Okay. The next different question is protein A from Pseudomonas petita contain 135 amino acid. Okay, here the rule is that they are asking us to find number of nucleotide present in the gene encoding the protein. They are asking how many nucleotide sequences are there in the mRNA. Okay, the formula to find the length of mRNA is equal to number of amino acid into 3. So 135 is the number of amino acid multiplied by 3, you will be getting 405. Okay, but this 405 is the final answer. They are asking number of nucleotide. So number of nucleotide present in the gene is equal to 405 plus 3. What does this 3 means? This 3 is the stop codon. The stop codon is made up of 3 bases. This 405 bases will be coding and it will be translating to the protein. So here their question, they are asking how many number of nucleotide is present. In that case, 405 plus 3 base pair. This 3 nucleotide includes the base pair of your stop codon. So what is the correct answer? 405 plus 3, which is 408. That is, you need to find the difference between this question and you need to find the difference between this question. Okay, next the DNA content of the cell in G1 phase of the cell cycle is C, what will be the content after meiosis is completed. So whatever the theory kind of thing you have, just keep it aside. So just remember the formula sheet for mitosis. I had given the N is the number of chromosome and C is the DNA content. So number of chromosomes at various stages I had given and number DNA content at various stages I had given. For G1 phase, the number of chromosomes will be 2 and the DNA content will be 2C. Whereas for S phase, again the number of chromosomes will be remaining same only, but the content, the DNA content will be double because we had gone through a process called replication. So the number of DNA will be, the DNA content will be 4. In G2 phase also, the number of chromosomes remain same and the number of DNA content remains same. In Pro phase, the number of chromosomes is 2n and number of the DNA content is 4C. And in Meta phase, the number of chromosomes is 2n and the number of the DNA content is 4C. And regarding to anaphase, the number of chromosomes will be double because the sister chromatids will be separated. So at that case, the number of chromosomes will be double. So the number of chromosomes will be 4n and the DNA content will be remain same, that is 4C. Regarding to telophase, where there will be separation, one cells will be divided into two cells. Okay, in that case the number of chromosomes will be reduced or it will be coming into half, that is telophase. The number of chromosomes will be 2n and number of DNA content will be 2C. Same for meiosis also I had given. Meiosis, the G1 phase, it will be 2n, that is 2 chromosomes will be present, the DNA content is 2C. And in synthesis phase, the number of chromosomes remain same, but the DNA content will be double, that is 4C. And G2 phase also numbers remain same, and in prophase 1 of meiosis 1 also same. And in meiosis 2, that is metaphase 1 of meiosis 1 is also same. But in anaphase, what happens in anaphase 1? So meiosis 1 is called as a reductional division. Okay, so the homologous chromosome will be separated. So that's why the number of chromosome will be reduced. So this is different from that of mitosis. In anaphase 1 of meiosis 1, the number of chromosomes will be reduced. At the same time, DNA content will also reduce. Number of chromosomes will be n and the DNA content will be 2C. In telophase 1, the number remains same. Whereas regarding to meiosis number 2, where there will be a separation of your sister chromatids, so from 2 cells there will be formation of 4 different cells will be formed. So this meiosis 2 is similar to your mitosis. So the number of chromosomes in prophase 2 is n and DNA content is 2C, and in metaphase number of chromosomes will be n and the DNA content will be 2C, whereas in anaphase 2 the number of chromosomes will be doubled. Okay, the number of chromosomes will be doubled, it will be becoming 2n, whereas the DNA content remains the same. Okay, this is similar to your mitosis, where in mitosis the number of chromosomes is doubled when compared to the metaphase stage. So similar. So just remember this particular thing. So now the question is that the DNA content of cell in G1 phase of cell cycle is C. What will be the content of the DNA after the meiosis is completed? So the end of the meiosis, the number of chromosomes will be n and the number of DNA content will be C. So the DNA content will be reduced to half. The correct answer is C by 2 because in initial the G1 phase of cell cycle it is C. The DNA content is C. So after meiosis completed it will be becoming half. Therefore the answer is C by 2. Okay. So here if you take in this particular formula sheet the number of DNA, the DNA content at the G1 phase of meiosis is 2C. Therefore at the end of meiosis completed the number will be reduced. Okay. So the next question is which of the following is a palindrome? Okay, so for those children who don't know about this palindromic sequence, so I'll be discussing this particular question in the next video. Thank you friends, thanks for watching this.